Okay, so I start recording the meeting. Thank you very much for your presence today, all on this Zoom meeting. I could do a very classic introduction to Ruslan Medzhitov, but given today's audience, I think this is not necessary. I'm sure you all know that Ruslan was born in Uzbekistan, studied biology in what was then the Soviet Union, and then after some time in California, joined Charlie Janaway's lab at Yale University and made a major discovery about toll-like receptors, a work that played a key role in reshaping contemporary immunology. You also all know that Ruslan is a leading immunologist who has been the beloved mentor of many. I would like to say something different. Because we are a lab that gathers biologists, medical doctors, and philosophers of science, I would like to say why Ruslan has been a major inspiration for us. Ruslan is, for us, a paradigmatic example of a conceptual biologist. No doubt, Ruslan is also a fantastic experimental biologist. But precisely, Ruslan's work over the years demonstrates the absurdity of the all too frequent idea that in our times, biology should be all about experiments and theory-free data. When I interviewed Ruslan already 7 years ago at the Pasteur Institute in Paris, where we had invited him with Gérard Hébert, who is with us today, he said explicitly that what drives his research is the exploration of new concepts. This is also very clear if you read the remarkable interview published in 2011 under the nice title, Innovating Immunology, an interview with Ruslan Medzhitov, where Ruslan says, the idea is to find a question that is not obvious in the field and that addresses a conceptually novel question, and ideally something that is simple, end of quote. And if you check carefully Ruslan's recent papers, you will certainly be struck by the fact that 3 of his main aims are, first, to use the power of concepts to reorganize pre-existing data in novel and fruitful ways. 2, to undermine some of our most unquestioned scientific assumptions. 3, to create bridges between disciplines, for example, between immunology, ecology, and evolutionary biology. These 3 aims are typical of what we call philosophy in biology and medicine, and that constitutes the foundations of our philosophy in biology and medicine network, which promotes productive collaborations between biologists, medical doctors, and philosophers, a network of which Ruslan very nicely accepted to be a member. So Ruslan is much more than an immunologist. He is a thinker of immunology, or even better, a thinker of biology with a strong interest for different roles played by the immune system in health and disease. For all these reasons, we are extremely happy and honored to welcome Ruslan Medzhitov in this Fin in Biomed seminar. Thank you very much, Ruslan, and now the floor is yours.
Thank you very much, Thomas, for a very generous introduction. It's hard to give talks after such a such a generous introduction. And it's my pleasure to talk to you today, and especially given your interest in conceptual aspects of biology, and I will try to combine today some of the conceptual frameworks that we're thinking about with some experimental work. So I'll talk about, broadly speaking, about relation between homeostasis and inflammation, because I think these concepts are fundamentally interrelated, and it's hard to understand inflammation, or impossible to understand it without knowing what how it connects to homeostasis. And I'll try to, one of the themes of my talk would be the relation between concepts that have been developed in different fields. And I think that's something that more of that in biology, I think, would be helpful to adopt and and utilize concepts that have been developed in other fields where we may not know that they are relevant for questions we are studying, because biology suffers from lack of appropriate formalism and abstraction. I think that's a major limitation. So I'll give a brief overview of inflammation first, and then the first part of my talk would be about some of the ideas, and then second part would be about some of the data that experimental data that were guided by these ideas. So the origin of the modern concept of inflammation goes back to about 100 years. Rudolf Virchow and Leonid Medzhitov were probably the most prominent contributors to this concept. And the reason I wanted to start with this photo is because they represented two perspectives on understanding biology of inflammation. Virchow, of course, was proponent of a view that inflammation is a pathological process, and Medzhitov was one of the first people who recognized that inflammation has beneficial roles, and that there is a whole spectrum of inflammatory responses. And this idea is captured in this diagram that I call Medzhitov's triangle, because it's conceptually very deep but underappreciated. What Medzhitov thought of inflammation is that it's being on the spectrum, where at the basis is what he called harmony-disharmony balance. In modern terminology, this is what we call homeostasis, but the term homeostasis hasn't been coined yet at that time. But that's exactly what we would call homeostasis. Then he thought the next on the spectrum would be physiological inflammation, and then pathological inflammation, and then immunity to infection. So this would be a spectrum of conditions. And again, mostly since then, physiological inflammation in relation to homeostasis have been largely ignored, and most focus has been on pathological inflammation and role of inflammation in immunity. And that is a huge oversight, I think, and that's something that's now slowly getting modified, and now we more and more in the field, people appreciate that inflammation plays various physiological roles, and including in metabolism and other processes. But we still don't have the biological framework to incorporate inflammation, physiology, homeostasis, and pathology. So one way to think about, so the relation between homeostasis and inflammation may seem paradoxical, because these are kind of opposite states of the system. Just to remind you, homeostasis generally refers to maintenance of stability of biological systems, whereas inflammation is induced when perturbations are too great to be handled by homeostatic mechanisms alone, when homeostatic capacity is insufficient to deal with perturbations. And that would be the case at the extreme in the case of infection and injury. But those are extreme cases, and it's important to recognize that there is a spectrum of conditions that can trigger inflammation. And inflammation operates at the expense of homeostasis to enforce its restoration. And that's the part that sounds very counterintuitive, and I'll try to explain it, because that's I think is the basis for pathological potential of inflammation. So we can think of homeostasis as symbolically here as homeostasis operates based on negative feedback. When this ball represents a system state, if the state deviates from the desired state, negative feedback mechanisms will return it back to a normal or desired state that would be defined by some homeostatic set points, for example. But when these deviations go beyond what can be handled by homeostasis, then we can have this situation where the system state can go over the hill, where homeostatic mechanisms would be unable to return it back. And that's when inflammation is normally turned on to force it back into the homeostatic normal range. And if the system is outside of the normal range, this would be, by definition, this would be pathological states. And inflammation normally would operate to return it back to normal state, but it has this potential of, so this would be protective inflammation, the intended type of response. But inflammation has the capacity also to push the system outside of the normal range. In the case of acute inflammation, that can result in severe illness. And in the case of chronic inflammation, it can put the system into an alternative stable state that would be pathological. So this would be chronic diseases would be characterized with this. So this is a very symbolic way to look at it, just to appreciate the idea of relation between homeostasis and inflammation and stability of the systems. There are many different types of inflammation, depending on what induced the response. The classical conditions inducing inflammation are infection and tissue damage. For each of the causes of inflammation, there is a corresponding physiological purpose of the response, and then there is a characteristic pathological sequela. So in the case of infection, of course, the purpose is defense from infection. Possible pathological outcomes of excessive responses would be collateral tissue damage and sepsis. Noxious substances can cause a different type of inflammation where the purpose is to expel the substances from the body, and this would be allergic inflammation, and at the extreme, this would result in clinical allergies. Response to tissue damage, inflammatory response to tissue damage promotes tissue repair, when excessive can result in fibrotic diseases and cancers. And finally, the modern lifestyle can also be a cause of inflammation. And of course, there is no physiological purpose for this type of inflammatory response because modern lifestyle is not natural. But it's a very important cause of chronic inflammation, and many modern diseases, including metabolic, cardiovascular, and all age-associated diseases are characterized by this type of inflammation. So all these different types of inflammatory responses can be summarized in terms of 4 universal components. There is always some inducer of inflammation, like pathogens, damage, or loss of homeostasis. There are sensors that detect these inducers, including macrophages, mast cells, and some sensory neurons, epithelial cells, and so forth. And when they detect the inducers, they produce a set of inflammatory mediators, including cytokines, bioactive amines, eicosanoids, and so forth. And these act on a variety of target tissues, including effectors like neutrophils and monocytes and eosinophils and so forth. So these are 4 universal components. And this actually represents an example of a control circuit that is much more generic than just the inflammatory pathway. Exact same control circuit operates in homeostatic pathways, as illustrated here. It could be either engineered or biological circuit. Here's a familiar example with engineered circuit would be a thermostat connected to a furnace or air conditioning. And the way they operate in either engineered or biological systems is based on the exact same principle. There is some variable that the system cares about, like in this case, it would be room temperature. If it deviates from the desired value, the sensors will detect deviation, produce signal that will act on effectors that can correct the deviation. The effectors here will be furnace or AC, and they will correct the deviation from the desired value of regulated variable. So that's formal description of how homeostatic circuits work as well. So familiar example in systemic homeostasis would be deviations of blood glucose, for example, from the desired value of about 5 millimolar in adult humans. The sensors would be alpha and beta cells of the pancreas, signals would be glucagon and insulin, and effectors are always part of the circuit that can correct the deviation. So in this case, it would be liver and muscle and fat, and they will change the value of the glucose concentration in the desired direction, at which point sensors will stop producing the signal and the system achieves the desired state. And in the case of inflammatory circuit, the input here, what we call regulated variable in the case of homeostasis, where we want the regulated variable to be at the particular value. Inflammatory pathway is basically the same thing, but the desired value would be 0, let's say. So you don't want any pathogens or toxins. If they're detected, so deviation here would be basically anything above 0, so detection of any pathogen or toxins. Sensors will detect that. In this case, these are inflammatory sensors like macrophages and epithelia produce cytokines that will act on effectors. Again, effectors is part of the system that can deal with these deviations. Again, since that value here is 0, so that would just mean that you eliminate them so that the response can stop and system gets back to homeostasis. So there is an important connection between the two types of pathways. So this is homeostatic circuit, and this is an inflammatory circuit. Again, homeostatic circuit, the sensors are endocrine cells or sensory neurons of the visceral nervous system. And effectors would be different organs or tissues that are relevant for whatever the regulated variable is. And in the case of, and the signals connecting them are hormones, whereas in inflammatory circuit, the signals connecting sensors and effectors are cytokines. And the important link between the two systems is that cytokines, in addition to acting on effectors like neutrophils, monocytes, T cells, and so forth, they also act on homeostatic sensors and effectors. And this effect of cytokines on sensors and effectors follows a certain logic. The way that these cytokines would act on homeostatic circuits is basically the effect on sensors would be to change the gain of sensors, meaning how much signal is produced in response to a given amount of deviation from a set point. And the way they act on effectors is to promote or inhibit their function and to change their sensitivity to hormones. And so this is the basis of, like in a very simplified form, the basis of connection between the two circuits. And the reason for the connections has to do with different aspects of defense that I will elaborate on through my talk. It is always useful when thinking about very basic concepts, it's always useful to look at them from more than one perspective. The more basic the concept is, the more different perspective it usually has. Like if you think of example, like the Pythagorean theorem has hundreds of different proofs because it's such a basic theorem. There are many different proofs for it. In the case of homeostasis, there are two very different perspectives we can use that offer very different insights that are both critical and complementary. The perspective I already described is a control circuit perspective. There is something you want to regulate, some regulated variable. In order to regulate, you need a sensor. And in order to change it in desired direction, you need an effector. So that's the control circuit perspective. There's a different perspective, systems dynamics perspective, where the regulated quantity, regulated variable, would be the stock of the system. And to change it, you either increase inflow or you increase outflow. So this is the stock and flow model of homeostasis. It comes from systems dynamics science. So here, a homeostatic sensor would detect or monitor the value of regulated quantity and then will act on inflows and outflows to adjust them so that regulate quantity at the right level. And so this, the processes, this inflow and outflow is what operated by the effectors in this perspective. So these two are very different perspectives, but complementary and both equally important. So now if we use this systems dynamics perspective, again, regulated quantity here is monitored by homeostatic sensors that produce signals with hormones and neurotransmitters that regulate inflow and outflow. For example, if this is blood glucose, this would be gluconeogenesis or glycogenolysis, and this would be glycolysis and glycogenesis. So this would be increasing or decreasing the value of blood glucose. And so homeostatic sensors would be endocrine cells or hypothalamus. These are hormones, and that's how the system operates. In addition, the inflows and outflows can be controlled by inflammatory signals produced by inflammatory sensors, and they are activated in response to inflammatory inducers. And notice that inflammatory inducers themselves are uncoupled from whatever regulated variable is. So the goal of this process is not to adjust the value of regulated quantity, not to keep it at the desired value, but to alter it to serve the function, the purposes of the inflammatory response. So here, sensors would be macrophages and so forth, and this would be inflammatory signals. And the way that the inflammatory signals will affect these processes is often antagonistic to the way that homeostatic signals will affect them. Not only antagonistic, but it's also dominant. And the reason for that is because the priority of the inflammatory response is higher than priority of many homeostatic circuits. The third perspective we can use to understand this, and this one is largely, I think, overlooked in both in physiology and inflammation biology, is this perspective that the sensors, when they detect either environmental condition or its internal representation through homeostatic variable, the sensors not only act on effectors to correct this homeostatic variable, as I just described. So this would be the homeostatic circuit operating based on negative feedback. But the signals would also act on part of the system that is not the effector, meaning this part of the system doesn't affect homeostatic variable, but the purpose for this signal to these non-effectors is to adapt to the new value of homeostatic variable. For example, if this is blood glucose and this signal is insulin, it will act on liver and muscle to promote glucose consumption to lower glucose level. But in addition, insulin will act on pretty much every other tissue and cell type, even though they don't participate in maintaining homeostasis of glucose. And the purpose of insulin effect on those tissues would be to, is a feed-forward loop to promote adaptation to higher level of glucose, and other hormones will do it for other types of regulated variables. For example, insulin will promote anabolic functions. So this is an important distinction between the effects of sensor-derived signals on effectors versus non-effectors. So this effect on effect, when signals act on effectors, it results in negative feedback. So the system resists perturbation to homeostatic variable. When they act on non-effectors, it acts through a feed-forward loop to promote adaptation to change in homeostatic variable. And that theme goes beyond just homeostasis. It also applies to inflammation and applies to different ecological modes of adaptations of animals, as I will refer to in the very end of my talk. But the way it applies to inflammatory pathway is that, just like what we saw with homeostatic circuits, there is an inflammatory circuit where pathogens detected by sensors, they produce inflammatory mediators, they act on effectors like neutrophils and monocytes that then will result in elimination of pathogens. So this would be negative feedback, and this is what we would call resistance. But in addition, inflammatory mediators act on many other organs that don't directly participate in defense. And here they act again in a feed-forward manner to promote adaptation to this new state of the system characterized by the presence of pathogens and inflammation. And that part is what is much less understood and studied compared to resistance part. Resistance part is basically the antimicrobial response and immunity, whereas this part has to do with altering physiology in a particular way to promote adaptation and survival. And that concept relates to it's actually the same concept, but that goes by a different name that when it applies to antimicrobial strategies, survival strategies that are referred to as resistance and tolerance. So tolerance here is equivalent to the adaptation term I used earlier. And this idea is that the organism can deal with microbes in two ways. So if microbes are deleterious, if they're pathogens, low level of microbes is associated with a healthy condition. When there is an infection or colonization, microbial load goes up and health can go down as a result. And there are two ways that organisms can deal with it, either through resistance, getting rid of microbes, or through tolerance or adaptation, promoting health in the face of microbial load. So the microbes that the organisms will learn to tolerate this way can eventually become commensal microbes, and we have plenty of them, and we are happy to have them generally because we can tolerate them. And other microbes we cannot tolerate, and again, we can either get rid of them through resistance or promote some adaptation through physiological changes. During an infection, microbial load is only one problem. The other problem is the inflammatory response itself. An exact same concept would apply here, where inflammatory response, lack of inflammation associated with health condition, when inflammation is induced, health may go down, we feel sick, and we can either regulate inflammation, so it would be equivalent to resistance mechanism because you return it back to normal through negative feedbacks. And we know that this is very critical because if you don't have negative regulators of inflammation, it almost certainly will result to lethal inflammatory diseases. But the other way we can deal with inflammation is by increasing adaptation to the presence of inflammation, because you can't always just decrease inflammation to be able to handle it, because it will compromise your defense from infection, for example. So we also need ways to adapt to a given level of inflammation. And inflammation can be deleterious because it comes with a cost. And there are two types of costs of inflammation. There is the simple one is on the right. This is unintentional but unavoidable loss of function of tissues. This is basically collateral damage. So that one is something we don't want, but we can't avoid it completely. The second cause of inflammation is more interesting in a way because it's intentional suppression of incompatible functions that have lower priority. For example, inflammation will alter homeostasis in a way that serves the goals of inflammation, but at the cost of other functions. But those functions would have to be first incompatible with inflammation, and second, they have to be lower priority functions. But it's still a cost nevertheless, and a combination of these two costs is what can ultimately result in inflammatory diseases when inflammation is excessive. So the cost of inflammation can be very significant, as we all know, and most diseases are associated with inflammation one way or another. And one way we can think about how and why inflammation comes with such a high cost is by looking at this trade-off between cost and benefit for any biological trait. So if you plot benefit and cost here, any trait that has cost higher than benefit, which would be in this red triangle, would be eliminated by evolution. Any trait that has benefit higher than the cost would be maintained by evolution. And this is evolutionary perspective. So anything in the green triangle would be fine. Anything in the red would be eliminated. So from evolutionary perspective, anything here is fine. But from patient's perspective, the same trade-off looks like this. Because as you increase the cost in this area, in the red area, this is when you would have all the symptoms of inflammatory diseases or even acute infection. And even though benefit is still higher than the cost over here in this upper red part of the square, here it comes with very high cost because it provides very high benefit. And the high benefit, the highest benefit you can have is survival from infection, for example. Because inflammation can provide very high benefit, it comes with a very high acceptable cost. And so from patient perspective or doctor's perspective, this is what it would look like. So many so-called diseases and many inflammatory conditions initially were characterized as diseases because of that, even though they may have beneficial role to the organism's survival. So some diseases, in other words, are due to evolutionarily high acceptable costs of inflammation and other processes that have high costs. So we were interested in what happens in this corner of this square, where the traits like inflammatory response have very high benefit and very high acceptable costs. So here, if you're very close to this diagonal, you're basically standing one step away from pathology and from death, in some cases when it's very high here. And we thought there must be some mechanisms that evolve to deal with these conditions when you're one step away from death, and yet the system is designed to operate in that regime. And so one set of conditions that are known to operate in that state called sickness behaviors that are characterized by loss of appetite, social withdrawal, fatigue, and so forth. So this is something everyone experiences when you have acute infection, for example. And we were interested in how these sickness behaviors, how they're all induced by inflammation, and mostly through the effect of inflammatory signals on hypothalamus. And often it involves change in the set points that is set up by hypothalamus. But we were interested in how that operates in the context of acute illness and how it may be helping survival of the organism. So I'll show a couple of stories that are, I will shorten version of it, not going through all the data, just the data that are necessary to illustrate some of these points. So in the first story, we asked how loss of appetite, which is something that we all experience, can be very dramatic when you're very sick, you don't want to eat, and why that happens. And that's true for all organisms studied, not just for humans and vertebrates. It's true in insects and other animals. So we first started by repeating experiments that were done actually in the 70s, where we infected mice with listeria, and infected mice stopped eating as expected, and they dramatically, infection suppresses appetite. This could be mimicked by just giving them LPS, lipopolysaccharide, and that would cause systemic inflammation, and that would also lead to suppression of appetite. And then we asked what would happen if during this state, if mice are, if they would eat still, even though they don't have appetite, if we basically force feed them. And this is what happens. If you give them LD50 dose of listeria, where half of the mice die, and then if we either give them PBS, basically nothing, saline solution, or we give them food, the mice that received food, they all died. And we gave them only 20% of what they normally would eat, so they're not stuffed with food. And this is actually presumably good food, because this is the same food that's given to ICU patients, at least in our hospital. So they all died when they received food. And if we do the same with LPS, induced inflammation, and this model of sepsis, again, mice that receive PBS, this is a sublethal dose of LPS, most of this will survive, and mice that receive food, well, most of them die. Then we asked, what is it in food that makes this difference? And it turns out it's not proteins and lipids, it turns out to be carbohydrates, and specifically glucose. So just giving them glucose turned out to be enough in the context of systemic inflammation, was enough to kill them. And this is shown here with glucose giving either IP, or it could be given orally, didn't matter, it would still result in 100% mortality. In contrast, if we give them 2-deoxyglucose, which is anti-metabolite for glucose, that blocks glucose consumption metabolism by cells, then 100% of them survived. So it was very striking that such a simple modification, simple manipulation of animals could result in 100% life and death difference. These effects of glucose or 2-deoxyglucose that resulted in either survival or death, importantly, was not related to the magnitude of inflammatory response, as shown here. So inflammatory cytokines, acute phase proteins were induced to the same degree, whether they given glucose and died, or 2DG and survived. So it wasn't dependent on inflammation, and it was not dependent on, when we did it in the infection models, wasn't dependent on the microbial load. But what it was dependent on metabolic adaptation caused by inflammation and anorexia that switched from one type of fuel, from glucose, to another type of fuel, which is ketone bodies, such as beta-hydroxybutyrate. So the way we found it is that, so when mice are given infection or LPS, they stop eating, and when they stop eating, they switch on this program to produce beta-hydroxybutyrate that is necessary for survival of prolonged starvation. And if we delete this transcription factor, nuclear receptor PPAR alpha, that is necessary for ketone production, there is no ketones, and these mice die. So they cannot survive even without force feeding or glucose or anything like that. They would die if they cannot make this metabolite. So that shows an example of metabolic adaptation that is critical for survival. Has nothing to do with direct effect on microbes or resistance. So this is all adaptation. So the summary of this part, I'm not going to go through all the details, but summary is that in response to infection or inflammatory trigger, inflammatory cytokines promote metabolic changes, induce lipolysis, suppress appetite. Suppression of appetite results to drop in glucose, drop in insulin, and insulin is a brake on lipolysis and ketogenesis. As a consequence, beta-hydroxybutyrate is produced by the liver, and it's used by the brain as a fuel. And what we found is beta-hydroxybutyrate actually protects certain parts of the brain from inflammatory damage. And that's what was necessary for survival of acute inflammation. So based on that, we wondered whether, since surviving acute illness is clearly evolutionary would be very important process, and there may be specialized systems evolved to promote these physiological adaptations. We wondered whether sensors of inflammation, like macrophages, whether in addition to inflammatory signals that promote resistance mechanisms, whether there are specialized signals that have nothing to do with resistance perhaps, but primarily may be dedicated to promoting physiological adaptations. And such signals are not well known, not well characterized. Mostly we know about inflammatory signals. So we started looking for what might macrophages make in response to infection that may be dedicated to these pathways. And we got interested in this hormone called GDF15 because, for a couple of reasons. One, we found that it's very strongly induced in macrophages in response to microbes. And second, it's been implicated in a variety of pathological conditions, but it's induced by all kinds of stressors, including chemotherapeutic drugs, different types of oxidative stress, and other types of DNA damaging stress, infection, and it's produced in pregnancy also. And it causes appetite suppression, nausea, cachexia, and so forth. And a few years ago, because GDF15 has been known to be appetite suppressor, several pharmaceutical companies got interested in it. And this is, by the way, illustration how GDF15 suppresses inflammation if it's given to mice as a recombinant protein. And several companies identified a receptor for GDF15 that is actually expressed in part of the brainstem called area postrema, which mostly famous for as being kind of a vomiting center or nausea center. It's activated in response to various toxins and suppress appetite and so forth. So we thought, well, GDF15 is induced by inflammation, and it's known to be an appetite suppressor, so maybe it's responsible for this anorexia of acute illness, of acute infection. And so therefore we started studying it, and by the way, we found that it's actually not required for anorexia caused by infection. It's involved in other types of anorexia, but we found that it has some other interesting roles. So first we asked whether GDF15 plays any role in survival of acute inflammation, and we found that it does. So if we block GDF15 with neutralizing antibodies in the context of LPS-induced inflammation, you can see that these mice died, whereas control mice mostly survived this dose of LPS. Importantly, this did not affect the level of inflammatory response. So it wasn't suppressing inflammation. It acted in some other way. And again, remember, in the case of inflammation, two ways to deal with it, either to regulate it, to reduce inflammation, or to adapt to it. So this suggests that GDF15 promoted adaptation to inflammation rather than suppression of inflammation. And it was also necessary to survive bacterial infection. So this is polymicrobial sepsis, and this is flu infection. Blocking GDF15 in both cases resulted in increased mortality. Now, recombinant GDF15, on the other hand, did the opposite. So it promoted survival of these different forms of challenges, inflammation or infections, viral and bacterial infections. So somehow it was an important factor for survival, but it was not acting to suppress inflammation. So because the receptor for GDF15, or at least the only known receptor, is expressed in brainstem, we thought that perhaps it activates, regulates autonomic nervous system and controls metabolism. So indeed, area postrema shown here is activated in response to LPS, and this is dependent on GDF15. When GDF15 is blocked, this goes away, this activation. And somehow lack of this activation is associated with mortality. So it's important to activate this area of the brain. But to our disappointment initially, we found that it had no impact on blood glucose, fatty acids, or hydroxybutyrate, because we thought that if it affected by hydroxybutyrate, that would be linked to the previous story, and it would explain how it works. But it wasn't working by this pathway. And instead, what we found is that blockade of GDF15 resulted in increased cardiac damage. This is shown here by looking at biomarkers of cardiac damage, troponin 1. So it's increased in the context of LPS or poly-IC. It's a bacterial or viral inflammation. When GDF15 is blocked, this marker of damage goes up. And concomitantly with this, there was a reduction in cardiac output, and the animals that had GDF15 blocked could not maintain blood pressure and heart rate, and ultimately succumb to cardiac failure. And we found that the reason for that was not that heart rate is controlled by autonomic nervous system, and particularly sympathetic nervous system is important to maintaining high heart rate. But we found there was no difference in norepinephrine, which is a neurotransmitter of the sympathetic nervous system, no difference in the heart, in the plasma, or in fat, but there was a decline in the liver. So somehow lack of GDF15 resulted in reduced activation of sympathetic output to the liver, but not to other organs we tested. And concomitantly with this, we found that in blockade of GDF15 resulted in reduction in plasma triglycerides. We looked at all metabolites, and amongst the different metabolites, the one class of metabolites where we saw difference was triglycerides, which is all shown here. And so triglycerides in the circulation can have two sources. One source is the diet, and the other is the liver. So diet-derived triglycerides come in the form of kilomicrons, but these mice that we are studying, they receive infection or LPS, so they're anorexic, they don't eat. So this cannot be the source of triglycerides, and therefore it has to be liver in the form of VLDL. So these triglycerides in either type of lipoprotein particles are used by tissues in response to processing by lipoprotein lipase, which is a vascular enzyme that releases fatty acids from triglycerides. They're transported and used by parenchymal cells. And the reason I'm mentioning this is because for the next two experiments, we used an inhibitor of lipoprotein lipase to determine how GDF15 operates. The inhibitor is called poloxamer, so it blocks lipoprotein lipase, so triglycerides in the circulation cannot be consumed by tissues. And basically what we found here is that GDF15 controls hepatic triglyceride secretion, but not triglyceride uptake. And this is mediated by beta-adrenergic signaling of the sympathetic nervous system. So this is shown here. So this is, if you compare black and red graphs, Black graph is the control graph, and red is when GDF15 is blocked, and plasma triglycerides, you can see, are reduced when GDF15 is blocked in the context of LPS inflammation. However, if we give these mice isoproterenol, which is an agonist for beta adrenergic receptors, then triglycerides will be increased even if GDF15 is blocked. So meaning that adrenergic signaling operates downstream of GDF15. And blocking GDF15 had no effect on clearance of lipids. If we just give lipids to mice, they clear at the same rate with or without GDF15. And then we asked whether the effect of GDF15 required adrenergic signaling, and using adrenergic receptor knockout mice, we found that unlike wild-type mice where GDF15 increases plasma triglycerides here, in the absence of GDF15, recombinant, in the absence of adrenergic signaling, recombinant GDF15 had no such effect. So this shows both that GDF15 can increase plasma triglyceride and that it depends on sympathetic nervous system receptors for adrenergic receptors. And accordingly, mice could not survive the LPS sepsis, even if we give them GDF15, if they didn't have adrenergic receptor signaling, illustrating that GDF15, presumably through its effect on the brainstem, activates sympathetic output to the liver, leading to triglyceride production. And the remaining question was whether this, so we showed this part and we showed this part. The question is whether triglycerides in the circulation are necessary for cardioprotection. And that actually would be consistent with what's known about heart metabolism. Heart, unlike most other organs, actually, most other organs prefer glucose under normal conditions, but heart prefers fatty acids. So it would make sense if triglycerides were necessary, but we tested that by giving the mice intralipid administration. Intralipid is a mixture of triglycerides. It's something that's used in the clinic in critical care. So we gave mice, we blocked GDF15, and then we either didn't give them PBS, so those mice died as expected. But if you gave them just triglycerides, that promoted survival, and that resulted in decreased heart damage, indicating that indeed triglycerides were the downstream component. And basically, overall model is that when animals sense infection, not only they produce cytokines to promote resistance and various adaptations mediated by cytokines such as lipolysis, but in addition, they produce factors like GDF15 and presumably many others that act purely to promote physiological adaptations to this severe inflammatory state. And in this case, one mechanism by which GDF15 acts is through activation of sympathetic output to the liver, production of triglycerides, and cardioprotection. But that's one mechanism, and undoubtedly there are many other types of signals that operate like GDF15, but maybe acting on other aspects of physiology. And without a doubt, GDF15 has many other roles as well, not just the one that I described. So this, again, parallels two strategies of dealing with a problem, like an infection, defense, and adaptation. Could also be called resistance and tolerance. These are equivalent terms. In the case of resistance, effectors like neutrophils are activated to deal with bacterial pathogens. In the case of adaptation, signals like GDF15 and probably many others act on part of the body that are not effectors. They don't directly participate in getting rid of pathogens, but these effect of such mediators is important to promote adaptation and survival. And it's particularly important for vital organs like brain and lungs and kidneys and heart. And that's where this type of signals have their major effects. So in summary and in conclusion, I just want to get back to what I started with this general notion of how resistance and adaptation are two ways that any system can react to any perturbation. And to just summarize it and to kind of bring it back to some additional related concepts. That what is adaptation? Adaptation basically is when it's possible only when the system has some alternative states, and that it can switch between alternative states in response to external perturbation in terms, changing the environment. And the choice between resistance and adaptation depends on their relative costs. And interestingly, that resistance typically comes with energy costs and adaptation comes with functional costs. So that's something that needs to be thought through more and tested in various contexts more, not just in the context of infections, but the nature of the cost for these two strategies, that it may be different. And so finally, there is an ecological counterpart to everything I discussed. And again, it comes with different terminology. And that's why biologists often study something, same phenomenon, and they don't realize that it's the same phenomenon that somebody else studies because they have different names. So in ecology, there is this concept of regulators and conformers. And what that means is that all animals adapt to their environment in one of two ways. Either they maintain their internal environment, which would be through homeostasis, or they may not use homeostasis to maintain environment, and they don't maintain it at all. They just conform to the external environment. And in ecology literature, these types of strategies are referred to as, the animals that use one or the other strategy are referred to as regulators or conformers, respectively. And importantly, the vast majority of animals are conformers. Regulators is more of an exception. So systemic homeostasis, as we know it, maintenance of internal environment, which is what regulators use at the organismal level at least, is relatively rare. And this is in contrast to what any medical student or physiology student would think, because most of the physiology that we study is based on mammalian physiology. But people who study more broadly animal physiology can tell you that, well, homeostasis as we know it in humans is relatively rare in terms of the majority of species. So this is where regulation and conformers. The simplest example is with temperature regulation. So of course, mammals use thermoregulation. So this is, if we plot internal state versus external condition. So if we change the temperature, regulators can maintain it within a certain range. Whereas conformers, like a fish, the temperature of the fish, the body temperature, will change with temperature of the water. So water here is a regulator in terms of temperature, and this bass is a conformer. And it's not only true for temperature, it's true for metabolism, it's true for basically many aspects of physiology. And so as you can appreciate here, what do we call this strategy? This is equivalent to what we refer to as a resistance strategy. And what is this strategy? It's an adaptation strategy. So this works through negative feedbacks and homeostasis, and this works purely through adaptation. And so this is my final slide. Ultimately, this is related to yet another perspective that I think also lacks maybe a little bit of clarity in terms of the terminology. Robustness and resilience often used interchangeably, but I think they reflect different properties of the system. So here you can have two plants dealing with strong winds. The way that this tree will deal with wind versus this grass is very different. The tree will resist change in shape in response to wind. So it has feature of robustness. Robustness is defined by how hard it is to change the state of the system, whereas the grass, of course, will bend in response to the wind. And but it's resilient. It can return back to original state, and resilience can be defined as by how easy it is returned to original state if it's perturbed. And of course, robustness is good in some contexts, but if the environmental effect is too strong, so these systems are hard to change, but once they change, it's hard to get back. So it will be just broken tree, whereas this system's easy to change, but they're easy to return back. So this would be resilience. And finally, if you think about it on the spectrum, on the left side of the spectrum would be robustness, something that's very hard to change, but very hard to repair. In the middle would be something that's resilient, easy to change and easy to repair. And on the far right side would be something that we could call adaptation, which is easy to change, but the change is, it's a change of the state. So there is an alternative state of the system, unlike resilience. I couldn't find an example using plants to illustrate that, but this would be something like having a plant that is too short to be bothered by wind. So finally, environments can be hostile for two different reasons. For example, environments can be hostile for multiple different reasons. Could be pathogens, predators, temperature, lack of food or water, and so on. But all these different reasons can be grouped into two types. Environment may have something we don't want, like pathogens, or it may not have something that we need, for example, food and water. And both types of hostile environmental factors can be dealt with by resistance or adaptation. And that leads to 4 possible combinations of strategies. And that's something that would be interesting to explore in the future, which combinations are dominant in nature and why, and what dictates the choices of different strategies we use to deal with hostile environments. So finally, I just want to thank people who did the experimental part of the work. Andrew Wang, Harding Nguyen, and Sarah Nguyen were main drivers of the experiments I showed, and Brandon and Fernando helped with various aspects of these projects, and this is my funding sources. And I'll stop here, and thank you very much for attention.
Thank you very much, Ruslan. That was very interesting. That was great. We had 3 questions on the chat, so I'm going to start with those questions and then ask everyone, if you want to ask a question, please use the participant option and raise your hand. It's going to be easier to handle. Because there are so many people today and we don't have that much time, I would ask you to ask very short questions if possible. So Ruslan, a question by Alexandra during the talk was, why does the priority is on the inflammatory signals rather than on the hormones?
So the reason for that is, so first, it's not generally true. It's true for the priority is determined by, the hierarchy is determined by which response has higher biological priority. Biological priority is defined by survival and ultimately by reproductive success, but in this context, survival is a good proxy for it. So if some process has stronger impact on survival, it will have higher priority. So the familiar example would be fight or flight response versus rest and digest response. Of course, fight or flight response has higher priority because it allows you to survive predators, for example. And digesting and the food would be lower priority because it's not essential for survival and will come at the expense of, of fight or flight response. The same idea applies to homeostasis and various homeostatic processes that have lower priority.
Okay, thank you very much. The second question was, what is the mechanism of lethality caused by Listeria infection that can be protected by BHOB and exacerbated by glucose? Is it mediated by the brain?
Yeah, so in this case, we know the answer. So what we found is that in the context of bacterial infection, if mice are given glucose, or if they force fed, what happens is that there is a region in the midbrain that contains neurons that are inhibitory neurons that have high metabolic rate. So if they consume glucose in the context of inflammation, that leads to their mitochondrial damage and ultimately neuronal damage. But if they are using ketones instead of glucose, then they're protected. And that's the basis for this mechanism of protection.
Thank you. One final question that was on the chat, and then we can go to the more direct questions, so to speak. Do you think the infected host senses the starved state in addition to sensing invading pathogens to orchestrate the required metabolic adaptation? So is there a sense of starvation or starved state?
Yes, for sure there are mechanisms that detect fasted or starved state, and they also translate into various physiological adaptations, including lipolysis and ketogenesis. However, interestingly, in response to when anorexia is caused by infection or inflammation, and animals also don't eat, their physiological and metabolic state is actually distinct from animals that would be just starving. So if you're just fasting or you're not eating because you feel sick, metabolic consequences are different. And that is very interesting because it's not, we don't know all the mechanistic details of how they're different, but we know that they're different.
Thank you very much, Ruslan. So I see that some people sent questions on the chat. So I suggest we take some direct questions and then we go back to the chat if this is okay for everyone. So I saw that Kama has a question. Kama, are you here?
Yeah, I'm here. Thank you very much for this beautiful lecture. So I'm just curious about more immunology question. Dear Ruslan, do you know what type of macrophages involved in this adaptation? Are they tissue resident or maybe some kind of microglia, because this is in the brain, so it will be like, I don't know, faster than other macrophages. So what do you think about that?
I think it depends on what kind of model one uses, but I think generally it would be tissue-resident macrophages that first detect the infection. Mm-hmm. So the answer would depend on where infection came from, but typically it would be tissue-resident macrophages that would be the main sensors and main source of inflammatory signals and GDF15. But how different types of tissue-resident macrophages might differ in this regards, and it's an interesting question that we don't know yet. That would be important to see if there are organ-specific responses depending on the types of tissue-resident macrophages in different organs.
But may I ask second question then? Then can we say that like tissue-resident, maybe they are more for adaptation, whereas the myeloid cells which infiltrate, they are more for resistance, like different functions of myeloid cells in this case?
I think in some sense, yes, there would be that trend. Tissue-resident would be participating in more homeostatic functions and adaptations, but they can participate in resistance as well. So I think generally they can do both, but inflammatory cells presumably would be more dedicated to resistance, and whereas resident cells more to adaptation. Yeah, I agree.
Mm-hmm. Thank you.
So please, if you want to ask a question, please use the participant option and raise your hand. I saw on the chat that Maya Saleh had a question. Maybe Maya, you want to ask your question directly, please? Maya, you're still here?
Yeah, I'm here. Yeah, my question was related to the cellular metabolic changes that occur in a macrophage after an infection, and then lead to epigenetic changes and the trained immunity process. Do you think this is an adaptation? And what's the cost? Is it an energetic cost in this case or a functional cost?
In the case of epigenetic changes in macrophages, I think those changes, they fall in both categories. There are some of these changes definitely associated with resistance, where, for example, antimicrobial genes modified in a way that increases their sensitivity to signal. But there are also various adaptation changes associated mostly with metabolism as far as currently known. How they associate with physiological adaptation, I think, is not clear yet. But generally, I would say both types of changes occur. So some of them resistance, some adaptation mediated by epigenetic changes.
Another question is by Jean-François Moreau. I can ask it because it's very short. You spoke about acute stressors, but what about chronic stressors? For example, aging.
Yeah, so with aging specifically, I'm afraid that the answer is that we may not have any dedicated responses to aging, and one can, because it's not something we expected to experience, and perhaps it could be also, the answer could be flipped, that because we don't have some of these dedicated responses, some aspects of aging are a consequence of that. But with other types of chronic challenges, definitely is an interesting question. Because the response to chronic challenge is very different from acute challenge, much harder to study, much more time consuming. So we don't know as much about that. But I would just generally answer that in any kind of chronic condition, chronic disease, there must be some perpetuating mechanisms that maintain it. Either some alternative stable state or something of that sort. And I think it's critical to understand what those mechanisms are to deal with chronic conditions, because chronic conditions generally are much more prevalent in modern world. But with aging, I'm afraid the answer is probably that there is nothing that specifically dedicated to dealing with it. I think the only mechanisms that exist are the ones that delay it or suppress mechanisms resulting in aging.
Thank you. I missed a question by Alexander Tkashev. Are you here, Alexander? Do you want to ask your question? Oh, in fact, you're here twice, I can see.
Yeah. Do you hear me?
Yes.
Yes. Nice to meet you. Yes. Can we trust? My question is, I'm a neurologist and I do a study about herniated disc resorption. It's a process that's driven by inflammation. So, can we translate the theory to the herniated disc resorption process? Because we have the macrophages, we have the cell stress effectors, and we have a result of inflammation, the healing process, the pure healing process from the beginning to the end. I'm sorry for my English, I'm a little bit in stress.
So I don't know how specifically it would, I'm sure generally it would apply in some level mechanistically. I'm not sure how it would apply. I just don't know how it would apply specifically to herniated discs, but generally tissue protection and is an important aspect of this entire sort of idea of tolerance or adaptation to damage. And many of these mechanisms I was referring to are mediated by increasing tissue protection. But with herniation is something, I think it's more of a important but more special case, which I don't know exactly how the adaptation mechanisms would apply there. And the healing mechanisms presumably would be similar to, the impact of these types of signals probably would be similar to other types of tissue damage, but I just don't know enough about the condition itself to answer more specifically.
One related question is you talked a lot about adaptation, and sometimes it seems like disease tolerance and adaptation are similar concepts, and sometimes it seems that disease tolerance is a subtype of adaptation. So how do these two concepts relate in your view?
I actually think they're the same concepts. Adaptation and tolerance, these are just two terms that apply to the same thing. And in the case of environmental adaptation, conformity would be yet another term that basically similar conceptually. Adaptation and tolerance, I think they're basically equivalents. They're not, they mean exactly the same thing.
Okay, so it means that we could not have a tolerance mechanism in a given organism that would be non-adaptive. That's not possible.
That tolerance mechanism that's not adaptive, yes. Yeah,
okay.
I would agree with it.
Okay, there's a question by Philippa. Can you envisage a situation where the non-effectors that drive adaptation can also feedback and talk to effectors that are responsible for resistance?
Yeah, that's a interesting possibility. I think presumably, yes. There is still very little known, of course, about these types of connections, but I would expect that, yes, there would be found situations where this that non-effector organs can affect the effector organ functions. There probably even something already known in the literature that we can interpret from that perspective. But yeah, I think there must be such connection. I'll just give one example, simple example where one can expect that to happen. When inflammatory signals act on non-effectors and change their functional state, for example, liver or kidney, each organ can handle only so much of deviation from the normal functional state. And once it reaches some critical value, presumably they will start signaling through negative feedback mechanisms to say, okay, that's enough, we need to, now we are becoming higher priority, because if there would be failure of the function of an organ, that also can result in catastrophic consequences. So yeah, I expect it would have, these types of communications would be identified.
One question by Tanya to follow up on the question about chronic inflammation. Do you think some inflammation-driven adaptations are always intended to be eventually reversed to the previous steady state? Are there adaptations meant to permanently switch the organism to a new steady state?
Yeah, I think generally I would say yes, they're always meant to be reversed, generally. The only hesitation with just saying that it's always the case is that some inflammatory, there's some processes where it's hard to draw the line whether they're homeostatic processes or inflammatory processes, because many inflammatory mediators participate in physiological processes not related to defense or anything like that. And in those cases, we can't really say that they're also meant to be reversed. So that would be the only exception. But generally, in the more sort of typical context, yes, I think they're always meant to be reversed.
Very good. I see that Alexander has another question. Maybe it's better to give the opportunity to someone else to ask a question. If not, I don't see any other question right now. So maybe Alexander, you want to ask your second question about the past? I don't see what the question is. Oh yeah, and there's, oh yeah, sorry, Alexander. There was also a question I saw before, but I missed it, sorry. So Alexander first, then Alexandra, and then we'll stop, because I know Ruslan has been tied with many things, including politics recently. So we don't want to take too much of his metabolic energy today. Alexander.
Yeah, we studied in our past in our institutions that inflammation is something bad, and now we know that is not always, and the most of modern treatment have an anti-inflammatory effect. Yes, and is there a place for in the future for some pro-inflammatory treatment or drugs or whatever? I mean, not reduce inflammation, induce or modulate inflammation in different conditions.
Yeah, I think the answer would be yes. We generally think of like a major culprits in acute inflammatory diseases, or even in chronic ones, which is these major cytokines like IL-1, IL-6, TNF. So in many contexts, when the levels of these cytokines are very high, would be associated with pathology and something we want to suppress. But there are more and more conditions found where additional types of cytokines that have roles that are more, they're inflammatory cytokines, but they have more roles that are tissue protective or participate in repair. And even the cytokines like TNF and IL-6 have many roles that are adaptive roles. So if we can figure out what would be the right doses of these types of signals, if they're insufficient, for example, for whatever reason, then yeah, I would expect that they could also be used with beneficial outcomes. And the main difficulty would be that administering them systemically probably not a viable strategy. So that would be main limitation, because they would need to be delivered in local controlled manner in small enough quantities. But if that's doable, if technologies would allow that, then yes, I think there would be many situations where that could be a good strategy.
Thank you very much.
Thank you. We will end with a question which I'm afraid is going to be a little bit difficult, by Alexandra. How did Janeway change your thoughts of the immune system? So you're allowed, Ruslan, to answer quickly if you don't want to spend too much time on that. But I think this is an important question nonetheless.
Well, in short, at the time, the immune system, to me, seemed like very too complicated, didn't make a lot of sense. There are too many factors, too many types of cells, and everything was kind of a huge mess in my mind. And then the Janeway's thinking about it was very different. It was basically providing a simple framework that a lot of known facts could fit in and could be tied together and explained. So that was a major way that it influenced my thinking. I think that type of perspective is generally useful in many fields, not just in immunology.
Great. Thank you very much, Ruslan. That was a wonderful moment with you. I really want to thank you. We really appreciate it. And we were 134, so really something we had never experienced before. So thank you very much, Ruslan. That was great. And see you soon. Thank you.
Thank you, Thomas. Thank you, everybody. Bye.